James Brown was a famous musician in the 1960s. You've probably heard his music on a movie soundtrack or maybe a commercial. James used to call himself the hardest working man in show business, and I'm sure you're probably wondering what that has to do with the nervous system. Well, the human nervous system is probably the hardest working organ system in our bodies. Whether you're working out or hanging out, the nervous system is busy coordinating all of the functions of the human body, from secreting hormones to digesting food to thinking about what happened last week or running a marathon. The nervous system is in control. But how does it control all of these activities? It uses a sophisticated language of chemicals and electrical impulses that can send messages with speeds ranging between 268 miles per hour and about 1 mile per hour from the top of your head to the soles of your feet. In this video series on the nervous system, we'll look at how the nervous system is organized, and we'll peel back the curtain on how it does its work. By the end of this video, you should be able to describe the functions and organization, that is the divisions, of the human nervous system. You should be able to describe the functions of the cells that comprise nervous tissue, and you should be able to label the parts of a neuron and state their function. Finally, you should be able to briefly describe the 3 types of neurons. The human nervous system has 3 main functions. The first function is to receive sensory input from specialized cells called sensory receptors. These sensory receptors send information about changes to our external and internal environment to integration centers. Function number 2 is integration. Integration is the process where sensory input is interpreted and a decision is made about whether the body should react to the sensory input. On the example here, sensory receptors in the eye send electrical impulses or information about light and color to the vision centers in the brain. Integration of that information tells the brain that the eyes are seeing a cup of water. If the brain is receiving a thirst signal from a different sensory neuron, then it may coordinate signals to contract the skeletal muscles so that they'll reach out and pick up the glass of water for a drink. This is the third function, motor output. The integration center coordinates signals to respond to the sensory information and to cause some part of the body to start moving or secreting. Your nervous system is divided into 2 anatomical divisions. The central nervous system, depicted in blue on this figure, is made up of the brain and the spinal cord. Its main function is to receive information from various parts of the body and interpret it and determine what to do. It is the integration and command center for the body. The peripheral nervous system, which is shown in purple, is made up of all of the nervous tissue outside of the central nervous system, namely nerves, which are basically bundles of neuron axons, and we'll talk about what those are, and you have ganglia, which are little bundles of neuron cell bodies. Nerves can be placed into 2 groups. You have the cranial nerves that arise from the brain and brainstem, and then they go out to the rest of the body. And then you have the spinal nerves, which arise from the spinal cord and go out to the rest of the body. Both sets of nerves bring messages to the central nervous system and take messages from the central nervous system out to the rest of the body. So it's like a 2-way communication system. So here at the bottom of this slide, you can see a little flow chart that shows you the human nervous system breaking up into 2 parts. The one that we just talked about first was the central nervous system, and then the peripheral nervous system. Let's talk a little bit more about the peripheral nervous system. As I mentioned on the last slide, the central nervous system has one main function. It receives information, interprets it, and decides what to do about it. This is called integration. The peripheral nervous system is divided into two functional divisions. The sensory or afferent division receives information about what is happening inside as well as outside of the body, and it conveys that information to or towards the central nervous system. The somatic sensory fibers, which are listed here, convey information such as temperature, pressure, stretch, or location, for example, from the skin, joints, and skeletal muscle to the central nervous system. The visceral sensory fibers do the same thing, but for your visceral organs. These would be organs like your stomach, your liver, your lungs, and blood vessels. The motor or efferent division of the peripheral nervous system conveys information from the central nervous system to the muscles and other structures of the body. Somatic motor fibers stimulate skeletal muscles to contract and glands to secrete. Visceral motor fibers stimulate visceral organs like the heart and stomach to contract rhythmically. One important distinction between the somatic motor and the visceral motor system or division is that the somatic division is voluntary. So in the last chapters when we said that you consciously control the contraction of your skeletal muscles, this is the division that we mean. Other muscles, called smooth muscles, contract without conscious control. They are under the control of the visceral motor system. We also sometimes call the visceral motor system the autonomic nervous system. So the ANS that you see on this slide right here, the A stands for autonomic, and the N is nervous, and the S is system. The autonomic nervous system is further divided into two parts, the parasympathetic and the sympathetic division. Parasympathetic is in control when we're resting and digesting. It tries to conserve our energy, and it promotes the general function of the body. The sympathetic system controls many functions of the human body when we're active, embarrassed, or frightened. We call this the fight or flight response. It promotes us to get up and go quickly. The last thing I want to say about this slide is that the motor division is called the efferent division because electrical impulses from the central nervous system exit the central nervous system and go to the rest of the body. I've drawn the arrow here in gray to help you remember that signals are coming from the central nervous system and going out to the rest of the body for the efferent or motor division. Afferent, that's the sensory division, conveys electrical impulses and information towards the central nervous system. So I've drawn this arrow to help you remember that. We say that electrical impulses from the afferent division arrive at the central nervous system. So the A in arrive and the A and afferent go together, and the E in exit and the E and efferent go together. This flowchart puts the central nervous system and the peripheral nervous system together under the heading of the human nervous system. Although there are many ways to divide the nervous system in terms of structure and function, it's important to remember that both the central nervous system and the peripheral nervous system intersect in a normally functioning human body. The general flow of information is shown on this slide. So, sensory receptors located inside our body and on the surface of our body detect stimuli, that is, changes in our environment. That information travels via sensory neurons to the central nervous system, where it is interpreted and a decision is made of how to respond to that stimulus. The central nervous system directs the response of our body to the stimulus through electrical impulses that go from our motor neurons out to our effector organs and tissues. So the effector is the organ that's going to perform the response. It's the heart muscle that speeds up or slows down its contraction. It's the skeletal muscle that contracts or lengthens. It's the gland that starts secreting. That all of those actually would be examples of effectors. Now let's look at nervous tissue. There are two principal cell types in your nervous tissue. You have the neurons. These are excitable cells that transmit electrical signals. They are the main communicators in the human body. You can see them in this slide of spinal cord tissue right here. Then you also have the neuroglia, sometimes called the glial cells. They're the supporting cells that help maintain the health of the neurons. And if you look really closely in this slide, you'll see the nuclei for your neuroglia peppered all throughout the microscope field here. There are multiple types of neuroglia cells. You will be responsible for knowing what each of them does. This shows up usually as a matching question on the exam. So your neuroglial cells include the astrocytes, the microglia, the ependymal, the oligodendrocytes, the satellite, and the Schwann cells. Let's look at them in a little more detail. Astrocytes are the most abundant, versatile, and highly branched glial cells. As you can see here, they look a bit like a star, which is why they're called astro, which means star, and cyte, which means cell. They cling to neurons, synaptic endings, and capillaries, and you can see they kind of wrap around those structures. Here's a capillary right here. These cells help to support and brace neurons, like you see here, and hold them close to the capillaries. Now, substances that are diffusing through the capillaries have to diffuse through the processes of these astrocytes, so they help determine capillary permeability. They also guide the migration of young neurons in developing nervous tissue, and they control the chemical environment around the neurons, which is essential to their function. These cells are also participating in information processing within the brain. The microglia are small ovoid cells with spiny or thorny processes. They migrate towards injured neurons and phagocytize neuronal debris. They also phagocytize microorganisms. This helps keep the area around the neurons clean and free of pathogens. Ependymal cells range in shape from squamosal to columnar, like you see in this figure here. These cells may be ciliated, and they line the central cavities of the brain and spinal cord. Their main function is to help make cerebrospinal fluid, and they help circulate it as well. Cerebrospinal fluid is the special fluid that helps maintain the buoyancy of the brain, and it helps to protect both the brain and the spinal cord. In addition, it serves as a conduit for the flushing out of waste from your nervous tissue. Your ependymal cells also help to separate the cerebrospinal fluid from the interstitial fluid of your nervous system tissues. Oligodendrocytes are branched cells. They have processes that wrap around the axons of neurons in the central nervous system. These wrappings form what's called the myelin sheath, and they help speed up action potential conduction in the neurons. Schwann cells and satellite cells do basically the same thing, but in the peripheral nervous system. Schwann cells, also called neurolimocytes, surround the peripheral nerve fibers or axons of neurons in the peripheral nervous system, okay, and they form myelin sheaths there. They're vital to the regeneration of damaged peripheral nerve fibers, and they help speed up action potential conduction in those fibers. Satellite cells surround the neuron cell bodies in your peripheral nervous system. Now let's talk about neurons. Neurons are also sometimes called nerve cells, and they are the main communicators in the nervous system. These cells have special characteristics. While you're in the womb, they are mitotic, that is, they reproduce. But after you're born, they stop reproducing. We call them amitotic for that reason. And there's only a few exceptions to this rule. These cells are long-lived. They basically live as long as you do for the most part. So that means that some people will have neurons that are over 100 years old. They have a high metabolic rate, and they depend upon a continuous supply of oxygen and glucose in order to live and function. Their plasma membrane is unique in that it's excitable. It participates in electrical signaling, and it also helps to maintain cell-cell interactions as we develop. Now I'd like to go through some of the anatomy of your neurons. The most prominent feature that you'll notice is this. This is called the cell body, or the perikaryon, or sometimes it's called the soma. This is the biosynthetic center of the cell. If you look at this figure, you can see the nucleus is there, so that means that any DNA and RNA that needs to get made, it gets made here. You can also see that there's a large amount of endoplasmic reticulum here in blue. These are called Nissl bodies, okay, and it's rough ER, so that means that protein synthesis is going to happen here. You have a well-developed Golgi apparatus. Again, this is involved in producing proteins and modifying them for transport within the neuron and for secretion. So one of the products that your neuron makes is those neurotransmitters. Some of those are protein-based chemicals, so it's important that this cell has the ability to make those components and then secrete them. So that's what the Nissl bodies do, and the Golgi is involved in. You have a network of neurofibrils that help to maintain the shape of the nucleus, of the cell body. And one structure that you'll want to take a note of is that the cell body tapers down to a funnel here. This is called the axon hillock. And the axon hillock is where action potentials are going to arise from. Inside of your body, you have clusters of cell bodies. They're called nuclei when they're located in the central nervous system, and they're called ganglia when they're in the peripheral nervous system. Next, we have the dendrites. You can think of dendrites as being like the ears for the neuron. This is where signals coming into the cell body usually come from. So these processes that are kind of extending from the cell body, these are the ears. They are called dendrites. Extending from the axon hillock down here, we have a long tail. This is called the axon. There's usually one axon per cell, and it always arises from the axon hillock. These axons can be very long. For example, there are some neurons that have their cell body in the base of your spinal cord, but then the axon for that neuron goes all the way down to your toes. So that means that they can be over a foot long. Sometimes you'll have branches, for example, arising from here, okay, from a neuron. These axons are called axon collaterals. So they're just branches from the axon. The axon is the conducting region of a neuron. So once an action potential is generated here at the axon hillock, it's going to race down the axon all the way to the end where we have these axon terminals. So in other words, they generate and transmit nerve impulses or action potentials, and they always move away from the cell body. The axon ends in a bunch of terminal branches. These are called telodendria, and each one has a knob-like axon terminal called a synaptic knob or a bouton. You can't really see them in this figure, but if you think back to Chapter 9 when we were talking about motor units, I had a picture that showed you a terminal branch and then a bouton. It looked like a button that was synapsing with muscle cells. These axon terminals are the secretory region of the neuron, and they release neurotransmitters to excite or to inhibit the activity of other neurons. The axon is insulated by a myelin sheath, and we find myelin sheaths both in the central nervous system and in the peripheral nervous system. In the peripheral nervous system, the myelin sheath is formed when a Schwann cell, like this one, wraps itself around the axon of a neuron, and it continues to wrap itself around, forming several layers. Okay, the lipoidal layer that you see here is the myelin sheath. Now, if you think about these cells as being kind of like the end of a tube of toothpaste, you know that if you twist the end of the tube of toothpaste up, eventually what happens is you get a fat bulge on the outermost round of the wrapped toothpaste tube. That would be equivalent to this. This is called the neurolemma. So as the Schwann cell wraps itself around the axon, the cytoplasm from the cell gets pushed outward to the neurolemma. The spaces in between the myelin sheaths, like this one, are called the nodes of Ranvier. So these are gaps between the myelin sheaths. This is where the axon collaterals can emerge. Some neurons are unmyelinated. Typically, these are very thin nerve fibers, and the consequence is that these nerve fibers transmit impulses much more slowly. Sometimes you'll have unmyelinated axons bundled together, so one Schwann cell can enclose about 15 or more unmyelinated axons. In the central nervous system, instead of Schwann cells, we have those oligodendrocytes. We will have nodes of Ranvier, but no neurolemma. The thinnest fibers in your central nervous system will be unmyelinated. So we're going to see this primarily with your thicker axons. Nervous tissue in the brain and spinal cord is classified as either white matter or gray matter based upon its appearance. So if you look at this brain from a cadaver, you can see that kind of the innermost or the deepest portion here has a very whitish cast. That would be white matter. And the outermost or superficial portion here has kind of a brownish gray appearance. So that's your gray matter. White matter is a dense collection of myelinated nerve fibers, and gray matter is mostly neuron cell bodies and unmyelinated fibers. There are three types of neurons based upon structure. The first is called a multipolar neuron. So the multipolar neuron looks a little bit like this. Where you have, this is your cell body, then you have several dendrites, like this, emerging from the cell body. And you'll have one axon, which is down here. Okay, so that would be your multipolar. Multiple dendrites, one axon emerging from the cell body. These are the most common type of neuron, and your motor neurons, which are responsible for moving your skeletal muscles, are this type. You have interneurons, which are neurons that are only found within the central nervous system. They're communicating. They are like relay stations transmitting impulses from one location to another. They'll also be this multipolar type. The second type of neuron is called bipolar. In a bipolar neuron, we're going to have a cell body like this, but there's going to be one dendrite and one axon. Something like this. Okay, these are relatively rare, and an example of a bipolar neuron would be your retinal neuron. So your retina is a layer in your eye that helps you to see. It absorbs light and transmits information about that light to your brain via the optic nerve. Third type of neuron is called the unipolar. This is a neuron like this, where we have a cell body here, and then we have just one process coming off of that cell body. And it has two branches like this. So your two branches, you have the peripheral process, which is the more distal branch, say this one. And then you have the central process like this one, which would be entering the central nervous system. So here are our pictures again of these types of neurons. Hopefully you'll recognize that this is your multipolar, this is your bipolar, and this is your unipolar. Okay, more pictures, just to give you an idea of the different appearances that they can have. So both of these are multipolar neurons, but they look very different. Both of these are bipolar, but they look very different. Unipolar, well, they all kind of look the same. Functionally, there are three types of neurons. You have the sensory or afferent neurons. They transmit impulses from your sensory receptors towards the central nervous system. So again, remember these are afferent. The impulses arrive at the central nervous system. You have the motor neurons, which are called efferent. They carry impulses from the central nervous system to your effector organs. The impulses exit the central nervous system. And then you have interneurons or association neurons. They basically shuttle signals. As I said, they relay information throughout the central nervous system, and most are entirely within the central nervous system. So here's some examples of your different types of neurons with respect to their function. So you'll notice here, this is a multipolar neuron. It has its cell body here in the spinal cord, and then it has a long axon that goes out, and the axon terminals synapse with muscle fibers. Okay, so this would be a motor neuron, and it conducts impulses along the efferent pathway. Some of your multipolar neurons are going to be interneurons. Okay, they are only within the central nervous system, kind of like this one. So they'll go from one place to another, either within the spinal cord or between the spinal cord and brain. Bipolar neurons, again, they're mostly sensory neurons located in special sense organs like in the retina, for example. So your bipolar neurons of the retina detect light, and then they convey that light towards the brain. Here's an example of a sensory neuron that is unipolar. So here's the cell body here, located in the dorsal root ganglion. Okay, you have the axon terminus here, okay, in the spinal cord, and then the lone dendrite is actually a sensory receptor located in the skin. So in this video, I've talked a bit about the functions and organization, that is the divisions of the human nervous system. We talked about, for example, the central nervous system versus the peripheral nervous system, and we talked about the somatic versus the sensory divisions of both systems. We also talked about the functions of the cells, the neurons, the microglia, the astrocytes, oligodendrocytes, Schwann cells, etc. We labeled the parts of the neuron and described their basic function, and we also talked about the three types of neurons based upon structure and function. All of this information is important for your exam. You'll want to be familiar with it in order to be successful in exam number 4. In the next video, we'll look at how your neurons actually conduct information to and from the central nervous system. I look forward to seeing you in that video. If you have any questions about this material or any other material that we go through, be sure to contact me either by email or text.